So once you have your libraries, you're ready for sequencing. Illumina sequencing is done on a device that's called a flow cell, and you can see it here on the slide. This is an old flow cell. There are newer versions available, and you can see them here on the next slide. I will come back on them. But the old ones are also still used. And as you can see here, a flow cell is just a glass slide with a metal grid on it. And that's the gray thing that you see here, that's the grid. So these glass slides is where you put your library on. The flow cell is put in the sequencer, and this is the surface on which the sequencing is done. So, and you see that the iron grid divides the flow cell into what is called lanes. So in this example, you see that there are 8 lanes. So all these transparent lines that you see here, they are lanes. And the number of lanes on the flow cell depends on the sequencer. So there are sequencers that use flow cells that only have 1 lane, but there are also sequencers that use flow cells that have 4 or 8 lanes. Every lane is further divided into what is called tiles, and that's what you see here. So here, these squares are tiles. And typically a lane is divided into 100 tiles. Now, on the slide here, it looks as if there is an additional metal grid that divides the lane into tiles, but that's actually not the case, so there's no iron grid here. The division into tiles is more virtual. So at some point during the sequencing, a camera will make pictures of the flow cell. So it will make pictures of every lane on the flow cell, but it cannot capture an entire lane on a picture. So what it does is it moves along the lane. So what happens is that the first picture that is taken will be of this part of the flow cell, so from this tile. And then the camera moves, and then it will take a picture of this part of the flow cell, so from this tile. Then the camera moves again, and it will make a picture of the third tile, and so on. So basically, a tile is the part of a lane that is captured in one picture that the camera makes. And during sequencing, for every lane, the camera will make exactly 100 pictures. So the newer sequencers, they use flow cells that look like this. They are called patterned flow cells. They are not glass slides, but you can see them as huge micro well plates because they have a pattern surface. They contain small wells in which the fragments of the library end up, and this is where they will be sequenced. Also, these flow cells are divided into lanes, typically 2 or 4 lanes. And also here, the camera cannot make a picture of the entire lane, so each lane is divided into multiple tiles. So for Illumina sequencing, the VIB nucleomics core uses either a NovaSeq sequencer, and this is one of the sequencers that uses these patterned flow cells. Or they use a MySeq sequencer, a MySeq Y100 sequencer that uses traditional unpatterned flow cells with only 1 lane for sequencing libraries from small organisms.